starting well. So, we have last session finished off with the topic of biotechnology, and today we go into applications of biotechnology, a chapter called as genetic engineering and genomics. Beautiful logic. Let's understand what do we exactly study in genetic engineering. So, सबसे पहले genetic engineering में we have a very good topic called as DNA fingerprinting. Now, what is DNA fingerprinting? DNA is the genetic material for most of the organisms, and though we inherit parental genes, we differ a lot from our parents mainly due to recombination of paternal and maternal genes. So, हम सबके अंदर DNA होता है and we resemble our parents, but there is a lot of variation between the parents and the offspring, and that is because of recombination of paternal and maternal genes. Differences also arise due to mutations which occur during cell division. Cell division में mutations होते हैं, उससे भी हमारे अंदर काफी variations होते हैं, and every individual has a unique genetic makeup which is called as its fingerprint. तो हर individual का DNA unique होता है और उससे उसका DNA fingerprint कहा जाता है. Now, the technique developed to identify a person with the help of DNA restriction analysis, also called DNA profiling, is also called as DNA fingerprinting. So, we identify a person with the help of a technique called as DNA fingerprinting, also called DNA profiling, also called DNA restriction analysis. And इसके अंदर एक बहुत important logic ये है, 99% of our nucleotide sequence in all people is the same. मुझ में, आप में, हम सब में 99% of our DNA is exactly identical, but 1% is different, and every person shows unusual sequences of 20 to 100 base pairs which are repeated several times. They are known as variable number of tandem repeats or VNTR. तो सर, हम सबके अंदर 99% DNA similar, but in that 1% difference that we have 20 to 100 base pairs का एक sequence called as VNTR, variable number of tandem repeats. और ये sequence called as VNTR is repeated in our DNA for 50% of our junk DNA. तो हमारा junk DNA, remember, 2% of our DNA is gene, 98% is junk, और इस junk DNA के अंदर continuously 20 to 100 base pairs का एक nucleotide sequence keeps on repeating again and again and forms 50% of our junk DNA. What are such repeating sequences called as VNTRs or variable number of tandem repeats? से मेरे DNA के अंदर मेरा एक सीक्वेंस है 80 80 80 80 80 80 GC 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 80 80 80 80 80 GC 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 GC, and this sequence is repeated again 80 80 80 80 80 GC 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 80 80 80 80 80 GC 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 GC, and repeated again. So that sequence is called as my VNTR. In your DNA, you may have your VNTR, which is 80 GC, 80 GC, 80 GC, CGTA, 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 and repeated again 80 GC, 80 GC, 80 GC, 80 GC, CGCA, 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 CGCA. Answer, that is repeated again and again. तो मेरा VNTR अलग है, आपका VNTR अलग है. Importantly, we all have 20 to 100 base pairs का a specific sequence of nucleotides called as VNTRs, which keeps on repeating again and again in our DNA and is called as variable number of tandem repeat or VNTR. Our idea of DNA fingerprinting is to exactly match those VNTR. तो उन VNTR को मैच करना है. How is that done? सबसे पहले VNTRs were isolated by Wyman and Weiss in 1980 at the University of Utah and USA, and probability of having similar sets of VNTRs in any two individuals is 1 in 300 million. So sir, two people having same VNTR, what is the chance 1 in 300 million people may have a repeat of their VNTR? So for all practical purposes, हमारे VNTR हमें unique है. Very impossible that somebody in the same area would be having your same VNTRs which are exactly matching your VNTR sequence. So सबका VNTR एक दूसरे से अलग होता है. The length of the regions having VNTR is different in each individual, hence is the key factor of DNA profiling. हम सबका ATGC sequence and length of our VNTRs are unique to us. Some people have 100 base pairs, some people have 20 base pairs, some have 30 base pairs, some have 40 base pairs, and ये सबकी unique VNTRs है. Length is different and ATGC sequences are different, and that is the way VNTRs are unique to us. Sir, what are the steps in the technique of DNA fingerprinting? Chalo, let's understand this. Sir, समझो you are a forensic expert and you go to a site of investigation. समझो एक जगह एक murder हुआ है and somebody was murdering a person. The victim just scratched the hands of the or the neck of the culprit. जो कल्प्रिट जो मर्डर कर रहा था, स्ट्रैंगुलेट कर रहा था या चाकू से मार रहा था, विक्टिम के हाथ उसके बॉडी के ऊपर आते हैं और उसके नेल बेड्स के अंदर यू गेट सम ऑफ द सेल्स ऑफ द कल्प्रिट. तो जिसका खून हुआ है उसके नेल बेड में खूनी के कुछ सेल्स आ जाते हैं. या समझो इट इज द रॉबरी. तो सर जहां रॉबरी होती है सर वहां पर वो रॉबर आया था, उसके सर से उसके कुछ हेयर फ्रैगमेंट्स गिर जाते हैं. एवरी डे वी लूज 5,000 to 10,000 हेयर. डोंट वरी, न्यू वंस डेवलप इमीडिएटली, बट हमारे सर से 5 से 10,000 हेयर के स्ट्रैंड नीचे गिरते हैं एंड वी गेट अ हेयर स्ट्रैंड इन द साइट ऑफ द रॉबरी व्हिच वाज बीइंग कंडक्टेड. Or suppose it is a rape case and the person who had raped the victim, he ejaculates on her और उसके semen के अंदर उसके sperms होते हैं. So sir, pick up one sperm, it has the DNA in the nucleus of the sperm and that can be isolated. So sir, first thing you do is do DNA isolation. DNA is obtained from any material like blood, semen, hair roots, tissue sample, so skin cells, hair cells, blood cells, tissue cells, कोई भी सेल अगर आपको मिल जाए से रेप हुआ है, a rape has taken place and from the vagina of the lady you take out some of the semen sample और उसके अंदर से आपको sperm मिलता है. So sir, that sperm has in it the DNA of the rapist, and then so you do DNA amplification. तो sir वो जो DNA मिला है उसका आप amplification multiple copies बनाओ. How do we make multiple copies of it? I hope you remember. Put it into a PCR box. PCR box, we just done that last session. Polymerase chain reaction is done and sir you will get multiple copies of that sperm DNA. तो स्पर्म डीएनए के मल्टीपल कॉपीज आपको मिल जाते हैं एंड देन वी डू डीएनए फ्रैगमेंटेशन. DNA fragmentation, remove one DNA and then you fragment it into small pieces with the help of a restriction endonuclease enzyme. And restriction endonuclease enzyme से उसके छोटे-छोटे fragments किए जाते हैं and each fragment has got a different length. The first fragment, say we use EcoR1. EcoR1 cuts GAA-TTC and EcoR1 cuts GAA-TTC and we get a sticky end. तो ये समझो आया first fragment, ये समझो आया second fragment, ये समझो आया third fragment. First fragment is large, second fragment is small, third fragment is medium, fourth fragment is very large. So sir, this means we have got restriction fragment length polymorphism. Restriction fragment के अलग-अलग lengths मिले हैं. Restriction fragment we got different lengths which is called as restriction fragment length polymorphism. Restriction fragment length polymorphism and sir ये डेटा आप कंप्यूटर में डाल दो कि सीमेन के स्पर्म में से डीएनए आइसोलेट करके उसका पीसीआर करके जब एक स्पर्म डीएनए को हमने कट किया तो उसका फर्स्ट फ्रैगमेंट वाज लार्ज, सेकंड वाज स्मॉल, थर्ड वाज मीडियम, फोर्थ वाज वेरी लॉन्ग एंड द एग्जैक्ट लेंथ इज फेड इनटू अ कंप्यूटर एंड दैट इज कॉल्ड एज restriction fragment length polymorphism. RFLP, restriction fragment length polymorphism. Then we do electrophoresis. फिर उसका क्या करेंगे? Electrophoresis. We take the fragments which are of different length and we subject it to a high temperature of 91 degrees Celsius. So 91 degrees Celsius temperature पे, I hope you remember, hydrogen bonds between the nitrogen bases will break and double-stranded DNA will become single-stranded. So double-stranded DNA single-stranded हो जाएंगे and single-stranded DNAs are then put into agarose gel. So I hope you remember, gel electrophoresis करेंगे. There are wells और wells के अंदर we put DNAs of different lengths, restriction fragment length, and we place a cathode here and an anode here, and we create an electric field. We charge it. DNAs will start going from the negatively charged cathode towards the positively charged anode. And you remember, this is called sieving. सबसे बड़े फ्रैगमेंट्स पीछे रह जाएंगे, सबसे छोटे फ्रैगमेंट्स आगे चले जाएंगे. So large fragments will remain behind, small fragments will go in the front और सबसे आगे सबसे छोटे fragments जाएंगे. By the way, which fragments are small and are kept on repeating again and again? If you remember, VNTRs. So variable number of tandem repeats are the most important. So समझो, this is your agarose gel and sir यहाँ wells है और wells के अंदर हमने DNA लोड किया तो यहाँ बड़े fragments of DNA आ जाएंगे and सबसे आगे कौन से fragments आएंगे? सबसे आगे आएंगे the smallest fragments which are the VNTRs. The variable number of tandem repeats are the most common and they will be repeated maximum times. So VNTRs सबसे ज़्यादा repeat होंगे and sir that will be done as a gel electrophoresis. Now that we have done this, what do we do next, sir? Sir, इस agarose gel के ऊपर we put a nylon membrane or a nitrocellulose paper. तो nylon membrane या nitrocellulose paper डाल दिया जाता है, उसके ऊपर टिश्यू पेपर रखते हैं Tissue paper is extremely hygroscopic. It will absorb all the water and the DNA fragments start sticking to the nitrocellulose paper or nylon membrane. And then we remove the nylon membrane or nitrocellulose paper, that is called as southern blotting. If you remember, this was called elution, and elution is also called as southern blotting. तो हमारे पास southern blot है, means बड़े बड़े fragments यहाँ नीचे है और सबसे छोटे छोटे fragments ऊपर है nylon membrane के. So this is your nylon membrane, and we have got the largest fragments below and smaller fragments above. And सबसे ऊपर कौन से fragments है? सबसे ऊपर we have fragments which are the VNTRs. तो सबसे ऊपर कौन से fragments है? VNTR, variable number of tandem repeats के fragments है. तो VNTR के fragments हमको यहाँ पर मिल जाते हैं. ये VNTR के fragments सबसे ऊपर है. This is called southern blotting. It is kept here. Now the police makes inquiry. They ask the lady, who do you think has raped you? She says, it was very dark. I couldn't see, but this person was sounding as somebody I know. So sir, she had not seen the face, but sir, she could identify according to the approximate height, according to the physical strength, that this person is known to her. And you will be surprised, in 90% rape cases, the identity of the rapist is known to the victim. So victim knows who the rapist is in 90% of the cases. So they have had some past interaction, and she says it was dark, I couldn't see his face, but I think it could be this person. And sir, तो पुलिस वो FIR investigation के लिए लिखती है, FIR किया जाता है, and all the people who are matching the description of the lady are called. तो sir, समझो 3-4 लोग आते हैं and they all are matching the description. सबसे पहले in a separate room they are made to sit down. The lady looks through a window to these people and she is told, can you identify by seeing who these people are? She says, no, it was too dark, I couldn't see them. तो सर ये 3 जन है या 4 जन है, चारों का सबसे पहले DNA आइसोलेशन किया जाता है. And what do we do, sir? Sir, we remove their buccal DNA. So they are told to open their mouth. We take a cotton swab and from inside the buccal epithelium we remove cells, and sir, these cells are taken. These cells are put into a cesium chloride solution. The DNA cell is centrifuged. DNA is isolated. I hope you remember by putting chilled ethanol, and this DNA is now cut with the help of restriction enzyme. Sir, in case of the first person, you find that the first fragment is small. The second fragment is small, third fragment is medium, fourth fragment again is small. If you remember, sperm का आपको क्या मिला था? Large, small, medium, very large. ये sperm का मिला था. अब इस first person का क्या मिला है? Small, very small, medium, small. तो restriction fragments, length polymorphism of the first person and the sperm DNA are not matching. So in all probability, this person is not the rapist. Then you check the second person, third person, and you find in case of fourth person, the first fragment is large, the second fragment is small, third is medium, fourth is very large. So he is having restriction fragment length polymorphism, sperm DNA and the fourth person's DNA restriction fragment lengths are matching each other. So restriction fragment lengths are matching. तो सर इस fourth person का पहले टेस्ट किया जाता है because restriction fragment length are matching, you put all these DNA fragments into a solution, raise the temperature to 91 degrees Celsius, break the hydrogen bonds, and double-stranded DNAs are taken, and double-stranded DNAs will become single-stranded. They are put into wells, place a cathode anode to gel electrophoresis. सबसे बड़े fragments पीछे रह जाएंगे, सबसे बड़े fragments आगे आ जाएंगे. Over it you put a nitrocellulose paper and you remove it. Southern blotting is done, and sir you find his smallest fragment. उसके सबसे छोटे fragment means his VNTR. His VNTR is now isolated. अभी बहुत ध्यान से इसके VNTR isolate करने के बाद उसको in vitro outside the cell we take this DNA fragment, his VNTR, and we prepare a probe DNA. तो ये है sperm DNA जिसका हमने जेल इलेक्ट्रोफोरेसिस करके नायलॉन मेम्ब्रेन पे सदर्न ब्लॉटिंग किया है, और ये फोर्थ सस्पेक्ट उसका हमने जेल इलेक्ट्रोफोरेसिस करके we got the small fragments and we pick up one fragment which is the VNTR. तो ये भी VNTR है, ये भी VNTR है, and we pick up the fourth fragment which is his VNTR and we make a complementary DNA parallel to it which is called as probe DNA. And probe DNA is made which is complementary to the VNTR of the fourth suspect. तो उसका probe DNA बनाया जाता है. And what is the probe DNA? It is a single stranded DNA which is complementary to the DNA of the suspect. तो उसका एक complementary DNA बनाते हैं, उसके अंदर isotopic nitrogen डाला जाता है. So what does the probe DNA have? Isotopic nitrogen N15 is used. If you remember, elementary is N14 and here what are we using? N15. So N15 isotopes are made against N14. The normal DNA is N14 and we are using N15 isotope, and we make a complementary strand of DNA which is called as the probe DNA. तो ये probe DNA बनाया जाता है. And now बहुत ध्यान से, this is probe DNA of the fourth suspect. We take this probe DNA and we put it into the nylon membrane, and nylon membrane के अंदर उसको डाल दिया जाता है probe DNA. बहुत ध्यान से, probe DNA, if it goes and gets attached to the VNTR, VNTR के सामने ये probe DNA जाके अटैच हो जाता है, which means this is VNTR of the sperm and this is probe DNA of fourth suspect. Probe DNA of fourth suspect. And this is probe DNA of fourth suspect. It goes and pairs with the VNTR of the sperm और दोनों के बीच में हाइड्रोजन बॉन्ड्स बनने लगते हैं. Then sir, it means that the sperm DNA, VNTR and the probe DNA, which is the VNTR of the fourth suspect, are paired with each other. And sir, ये हमको दिख तो नहीं रहा है. So what do? We put over it, I hope you remember, ethidium bromide. And ethidium bromide लेके उसको stain करेंगे, put it on an X-ray film. एक्सरे फिल्म पे डाला जाता है and we see it under ultraviolet light, which is called as photography or autoradiography. तो फोटोग्राफी और ऑटोरेडियोग्राफी, जहां डीएनए सिंगल स्ट्रैंडेड होता है, इट अपीयर्स एज अ थिन बैंड. Thin bands are seen wherever DNA is single stranded. तो thin band दिखाई देगा, but along with thin band, if you see a thick orange color band, a thick band is seen. Thick band is which one? Thin bands are single stranded DNA. Thick bands are double-stranded DNA, and this double-stranded DNA is a thick band, and this thick band is a clear-cut indication that the VNTR of the sperm and the VNTR of the fourth suspect are matching with each other. They are complementary to each other, and दोनों के बीच में हाइड्रोजन बॉन्ड्स बन गए हैं. तो अगर आपको थिक बैंड दिखाई दिया, that means the VNTR of the sperm DNA and the probe DNA of the suspect have paired with each other, which actually means that the suspect, this fourth suspect, is himself the culprit. Beautiful technique. What is this called as? DNA fingerprinting. इस तरह से किया जाता है. चलो, hope you all have understood this. And DNA fingerprinting is excellent. It is used in forensic sciences, and where else? Forensic medicine में use करते हैं. It is also used to establish parentage in disputed cases. Suppose a lady goes to a court saying, "This is my son, and this person is the father, but he has refused to take care of my child, does not acknowledge that child as his, and he is himself the father." The father person who is being accused, he says, "नहीं, I am not the father." तो उसका DNA fingerprinting किया जाता है, and you find that the DNAs of the father and this son are matching with each other. Their VNTRs match with each other, or their other gene fragments match with each other. Then we are able to establish a claim that yes, this person is actually the father, and then monetary rights are given to the child. The father is supposed to pay money for taking care of the child, and disputed parentage is now solved with the help of DNA fingerprinting. And also establish insurance claims or identifying a dead body. समझो एक person का एक dead body मिलता है. In Western countries, everybody's DNA profiling is done. So as soon as a child is born, उसका DNA profiling पहले से ही कर देते हैं, and then sir, this child grows up and there is a major fire in a building and you get dead bodies which are charred completely. तो उसके body के अंदर आपको कुछ भी दिखता नहीं है, the face is charred, so sir you cannot identify. Or you get a dead body from a lake and you find out the body has been completely swollen and mutilated. So you cannot find out the identity of the person by looking at the face. तो ऐसा burnt body या swollen drowned body, तो ऐसे bodies के अंदर जो उनके cells लिए जाते हैं, उसका DNA fingerprinting किया जाता है, and these DNA fingerprints are matched with the database of a person, and you find that the VNTR of the dead body and the VNTR of the database of the person which you have are matching, which means indeed that person जिसका database आपके पास है computers में is indeed the same person who has died over there. So identification of dead bodies also can be done with the help of DNA fingerprinting. And we have DNA fingerprinting में एक NCERT ने अच्छा स्टेटमेंट दिया है कि सर जब हम लोग VNTR और रेपिटेटिव DNA सीक्वेंसेस को आइसोलेट करते हैं तो सर यहाँ से एक वर्ड है सर when we do centrifugation you get two types of DNA. तो हमने समझो sperm DNA लिया है and we are doing a centrifugation, we get the DNA. We get two types of DNA, something called as bulk DNA. Bulk DNA are the larger fragments of the DNA. So उसको bulk DNA कहते हैं, which are large fragments. And which are the smallest fragments? The smallest fragments are the VNTRs. And VNTR, which are the smallest fragments, are called as satellite DNA. So another name for VNTR, variable number of tandem repeats, are also called as satellite DNA. So VNTR is also called satellite DNA. You have very small microsatellite and larger mini satellite. And सबसे छोटे VNTR are microsatellite, जो बड़े वाले होते हैं उसको mini satellite कहते हैं. And these are different names for the variable number of tandem repeats. तो इस तरह से DNA फिंगरप्रिंटिंग किया जाता है, जिसके अंदर VNTR and satellite DNAs are used for identifying the unknown person, and a beautiful technique called as DNA fingerprinting, which we have just understood. So good. So I hope you have understood this. We'll just have a small break and we go into another very important topic. I'll just see you soon. Take care.